Hello everyone, this is an introduction video for the pGLO transformation lab that we're going to be doing in the upcoming week. So with this lab, we're going to be working with the E. coli bacteria, and we're going to work with a plasmid that's genetically engineered. So this plasmid carry a pGLO gene that allows it to glow up if the environment for the bacteria is suitable. So this video is going to prepare you for the lab a little bit better. Let's take a look at this next slide. Here are some topics that we're going to be covering during this lab. There's genetics. So the most important part is genetics, DNA to RNA to protein and trait. But there are also, this lab also helps us relate to evolution and chemistry and cell biology, such as the cell membrane and how bacteria came about. So that's evolution. So that's DNA, RNA, protein, trait. So what is GFP? GFP is one very important part for our lab. The GFP is a visual marker that's naturally produced in some squid and octopus and jellyfish kind of animals. And the one that we're going to be using comes from a type of jellyfish. Scientists use it to tag cells or to make an entire organism glow, and it helps us track where a certain protein is expressed or how a gene expression is done within an organism. So we have the jellyfish right here, and then this is something scientists did to make fish glow up, and then we have the bacteria that can glow for this lab if we get it right this time. And how we use GFP as a biological tracer is we have our normal DNA, and if there is a certain protein that you want to track, you want to see where this protein is being expressed, or even where is this protein being made, and where does this protein travel to after it is made. So we can insert a GFP gene right at the end of the gene for the protein that you want to track. So then this protein is going to have a GFP marker attached to it so that this protein can glow. In this lab, I'm going to walk you through our procedure first. So what I already did for you is I incubated the E. coli bacteria for you at 37 degrees Celsius. So we're going to see colonies of bacteria. So if you can remember, when you see one colony of bacteria, that one colony came from one bacteria. So all the bacteria within that one colony should have the same genetic makeup. So what you're going to be doing is you will pick a colony that's big enough. It's about 1.5 millimeters. So you need a colony that's full grown in order to do this lab. What you'll do is you'll use this little tool to inoculate the bacteria and put it into two test tubes. One of the test tubes is going to include the genetically engineered pGLO plasmid that will allow this bacteria to glow, this E. coli bacteria to glow if it's put in the correct environment. And then you're also going to put the same bacteria from the same colony into a test tube without the pGLO gene. So that's our control. This one is not supposed to glow. All right, and then we incubate both of those tubes on ice for 15 minutes, and then we heat shock it at 42 degrees for 50 seconds. So this is what you need to remember when you're doing this lab. This process over here is very crucial for the success of our lab. So you really have to incubate it for exactly 50 seconds at 42 degrees Celsius. And then you put it back on ice for 2 minutes. And then we're going to incubate it at room temperature for 10 minutes. So your homework for Sunday is to read through the quick guide for the lab, just to make sure that you have a good idea on what's going to be going on in the lab so that you'll do it right. And then the next step is that after our plasmids, our bacteria is correctly incubated, you're going to plate it. So you'll put it on 4 different plates. There's going to be this one plate, LB is Lurea broth, so that's a nutrient for bacteria to grow. And I want you to think about, is the bacteria going to grow? And then we're going to put this same E. coli bacteria without the pGLO gene onto the LB AMP plate. And then you're going to put the E. coli bacteria with the pGLO onto LB AMP, and then LB AMP and the Arabinose plate. So the result is that the pGLO, the bacteria is supposed to grow on the LB plate because there's a nutrient on there, right? However, the bacteria without the pGLO is not supposed to grow on the LB AMP plate because the ampicillin resistant gene is only carried in the pGLO plasmid. So if the bacteria did not receive the pGLO plasmid, it's not going to have the ampicillin resistance, so this plate is not supposed to grow. And then we have the bacteria with the pGLO plasmid. So if a bacteria did take in the plasmid, it should be able to grow on the LB AMP plate because those bacteria are ampicillin resistant. And then the last one is the LB AMP and Arabinose plate. Arabinose is necessary to turn on the gene for the ampicillin resistant, and we're going to look at how that works in a second as well. So if the bacteria took in the plasmid and it has Arabinose to turn on the GFP gene, when we put this plate under the UV light, you're going to be seeing the glowing GFP bacteria. So that's all we're going to be doing. Tomorrow or Tuesday, we're going to do all these steps, and then a day or two from then, we'll be looking at the plates and analyze our results. So here's a visual representation on how the plasmid works. So remember, we have 2 pieces in our lab. We have the E. coli bacteria on its own, and we have the plasmid that's supposed to get taken into the bacteria for 2 of the plates. So this is the pGLO plasmid. We have an origin of replication to allow this plasmid to be transcribed and translated. We have the BLA gene, which is beta lactamase right here. And this gene, the protein that's coded by this gene, allows the bacteria to be ampicillin resistant if the bacteria did take in this plasmid. And then we have the green fluorescent protein, GFP, this GFP gene right here. And then we have the ARAC. So the ARAC produces a regulatory protein that allows the GFP gene to be turned on or off. So when Arabinose, the sugar Arabinose, is present, it's going to turn on this gene and make the regulatory protein that allows the GFP gene to be turned on, and then this GFP gene is going to make GFP protein, which will allow us to see the pGLO glowing. All right. So here we have the bacterial transformation picture. We have a bacteria and we have a whole bunch of plasmids that are ready to be taken in by the bacteria. So we're going to do the ice and then heat shot and ice again in order to make the bacterial membrane permeable for the plasmid so that the plasmid can be taken in by some of the bacteria. And we're going to see which bacteria takes in the plasmid by putting the bacteria onto the plate with ampicillin. So if the bacteria does grow, that means the plasmid has been taken in by the bacteria. So this plasmid itself can make the ampicillin resistant gene, and it can make GFP gene at the same time. So here's a picture that shows you how the Arabinose operon works. We have the ARAC, which is DNA binding protein, and we have the Arabinose. When Arabinose is present, so this is what the Arabinose operon normally work. This is not what we're dealing with, but this will help you understand the next picture. So this is what Arabinose operon normally works. We have the DNA binding protein, and we have Arabinose, and we have these structural genes that will make proteins to allow the bacteria to break down arabinose. So when arabinose sugar is present, it's going to bind to this promoter, pBAD, and it's going to change the shape of the DNA binding protein. After that, because this promoter is turning this part of the genome on, it allows RNA polymerase to bind to this gene more easily. So then RNA polymerase is going to be able to make these three proteins, ARAB, ARAA, and ARAD, to allow the broken down of the arabinose sugar. So what we did was this plasmid that we're using is, instead of having these three structural genes, we replaced it with a GFP gene instead. So this whole mechanism over here, where we introduce arabinose to turn on the gene, to turn on the transcription process, is still valid, except right now, instead of transcribing and translating proteins that can break down arabinose, we're making the GFP protein instead. Okay. So there are two things that are allowing us to have the bacteria taken in the plasmid. There's the electroporation, which is not what we're doing. So this is electrical shock that can make cell membranes permeable to DNA to allow bacterial transformation. But this is not what we're doing. What we're doing is the calcium chloride and heat shock. So the calcium chloride, right here, the calcium chloride gives you a positive charge to shield the negative charges of DNA phosphate. So it allows the DNA to be easier to get taken into the bacteria. And then we incubate it on ice to slow the fluidity of the cell membrane. And then we heat shock to increase the permeability of the membrane. So the membrane is now permeable to the plasmid, and then the plasmid can come into the bacteria. This is about Loria broth. Basically, it has a whole bunch of nutrients that will allow bacteria to grow properly. And I need you to, well, we already talked about this, but tomorrow and the day after, our key is to understand why exactly are we using the three different kinds of plates, and why we are using 4 plates. What results do we expect to see on those 4 plates, and what do we actually see, and what does that tell us? So your homework for today is to look over the quick guide, which is going to be sent to you later. The 2 extensions that we're not going to be doing is the GFP electrophoresis extension, and there's a chromatography. So for this one, we're not going to be doing it in our lab, but you can still understand it. So basically what's happening is we have this plate where the GFP gene is not expressed, and then we have this one where the GFP gene is expressed because we put arabinose in there to allow the expression of the GFP. And we can do an gel electrophoresis, and you can see the difference between the two, because the GFP gene itself is different from the genetic makeup. The genes that are produced here is different from here. So if we run gel electrophoresis for the proteins made, we can see the difference in the gel. And the one other really important one is this volume measurement. You're going to be using this graduated pipette. It's just a plastic pipette that's sterilized. And on your lab, you'll be able to see how you can measure the liquid that you're using this pipette. And we're going to practice using this pipette as well. The last really important thing is you got to make sure that everything is, all the tools that you're using, you have to keep those sterilized. So make sure that you're not touching any surface with the tip of the pipette, with the inoculation tube, and all of that. So this is it for our video. I hope that this will help you with your lab tomorrow.